So we finished up with talking about the cell wall inhibitors, and the next topic on the agenda are the inhibitors of the protein synthesis. So there are several major, six major classes. 5 of them are present over there. Okay. So we have 2 drugs that inhibit 30S subunit, and here 3 drugs are illustrated, but there's a little bit more. Just 4 different types of drugs that inhibit 50S. Okay. So 30 aminoglycosides. Remember, bless you. Remember we talked about Zellman-Waxman, how he discovered aminoglycosides, actinomycin, neomycin, streptomycin. Turns out when they bind the 30S subunit, which you see illustrated right here, they change the shape of the 30S, which doesn't prevent protein from being synthesized. But the protein that is made is incorrect. So those incorrectly produced proteins with the wrong sequence, essentially when they break down the match between the codon and anticodon. So say instead of serine there will be, I don't know, threonine inserted and stuff like that. So those mismatched proteins then can be transported to the membrane, can stop performing different functions, so they screw up the cell big time. Okay, and that was pretty much the first broad spectrum antibiotics, right? Tetracyclines that were discovered later are considered to be maybe the antibiotics with probably the largest spectrum. They can be used to treat virtually any type of microbe: gram-positive, gram-negative, obligate intracellular parasites, spirochetes. All of them can be treated with tetracycline, of course, with a slightly different level of success, but tetracyclines are used for pretty much all microbial infections. They prevent tRNA from binding to this 30S subunit, essentially stopping the protein synthesis. All right? So tetracyclines and aminoglycosides, broad-range antibiotics that bind to the 30S subunit. What about others? So macrolides prevent the ribosome from translocating, moving down the mRNA. Does that make sense? They bind to the 50S, so ribosome cannot move down, the mRNA cannot read the mRNA through, and protein synthesis is stopped. They're also static, have broad spectrum, and 2 antibiotics that you may have seen, very popular, erythromycin and azithromycin. That makes sense? Now, lincosamides. Lincomycin and its relative clindamycin modified version. So lincosamides also inhibit the formation of the bond between the growing chain of the growing peptide chain and a new incoming amino acid, as does chloramphenicol. Okay, so chloramphenicol is effective inhibitor, broad spectrum inhibitor of the peptidyl transferase activity. Essentially, it prevents the bond formation between the amino acid and the growing polypeptide chain. Right? And the newest of that group of protein synthesis inhibitors is Linezolid, that belongs to the group of the drugs called oxazolidinones. People to blame for such names are chemists. Okay, that's where it comes from organic chemistry. Okay, oxazolidinone is surprisingly a rather small molecule. Why I focus so much on linezolid? Because linezolid doesn't only have the broad spectrum of activity. It is quite an effective drug against the Mycobacterium tuberculosis. You will see a lot of time that I would say something. This drug works well against gram-positives. This drug works well against gram-positives and gram-negatives. And this drug works well against TB. Because Mycobacterium is notoriously hard to treat. I don't think we talked that much. It hides inside of macrophages. And that's one of the reasons. Okay. Now, linezolid is unique because when it binds to the 50S subunit of the ribosome, it actually prevents the ribosome from assembly. So protein synthesis doesn't even start. Does that make sense? These antimicrobials probably are the most used now in a clinical practice because they have such a broad range. They have fairly low levels of side effects because they have high specific toxicity. Okay, and a lot of them were discovered recently, so resistance to them is maintained relatively low. Does that make sense? Although, well, I must say some of them, like oxazolidinones, like linezolid, linezolid is considered to be a supplement drug, which means it shouldn't be used as the primary drug for treating any infection. Only if other drugs fail to treat the infection, linezolid should be used. That still allows to maintain the resistance levels to this drug at a fairly low. Now these guys, yes?
We were talking about types of delivery methods before. I know other drugs are given through inhalation. Is there any reason why antibiotics aren't given through inhalation, like for tuberculosis, for example?
That's a great question. Why? First of all, I think you will have problems reaching the concentration sufficient enough to treat. Second, Mycobacterium is not present as the free growing bacterium on the surface in the alveoli of the lungs. It is inside of macrophages, and the drug essentially has to be delivered into the macrophage. Another thing with Mycobacterium, as with pretty much any drug, it's easier to treat actively replicating microbe. Do you understand why? So when microbe replicates, it performs a lot of biochemical processes. It performs activity. It synthesizes the cell wall. It produces protein. You have something to inhibit. If microbe does not reproduce, if it's in a latent stage or an active stage, or call it whatever you want, you cannot treat it. You cannot treat endospore. That's another problem with Mycobacterium. If Mycobacterium doesn't replicate, and it replicates extremely slow, okay, it's really hard to treat. So I think the reason, so IV injections of the drug for Mycobacterium should be more effective, considering the circulatory network around the alveoli. Those drugs should diffuse fairly easily towards the site. And since Mycobacterium tends to go in acute cases, it can be extrapulmonary, so they can be neurotropic, osteotropic. That may also contribute, But inhalation, one of the major concerns with any type of inhalation drugs, concentration. Like with the vaccine that you probably have heard, the flu mist, one of the concerns about flu mist is that the amount that is delivered, with each spray is not, what's the word? Not the word reliable. It's not standard, okay? It varies greatly. So that's probably the reason. The answer? What did I just do? I wanted to chat just a few words about the membranes. I love these drugs. It's like nukes, okay, really, because they kill everything. Polymyxin, polymyxin B, polymyxin E, they work really well against gram-negatives, which is not very common. They work against multi-drug resistant, MDR, multi-drug resistant microbes, okay? Daptomycin is also good against gram-positive mostly, but also against multi-drug resistant. This picture actually shows you how polymyxin E, or colistin, binds to the membrane, okay, and disrupts its structure. Essentially, polymyxins just make membrane unstable. But as they can do it to the bacterial membrane, they can do the same to the eukaryotic membrane. Polymyxins are notoriously toxic. They are not used for other than topical treatment. You can see polymyxin B, topical wound infections, right? Polymyxin E, bowel decontamination. So it means that it's going to sweep through the bowels, killing everything there. The patient has nothing to lose at this point, okay? Systemic multi-drug resistant infections. So we talk about systemic infection with, I don't know, multi-drug resistant TB, okay? Multi-drug resistant gram-negatives. Colistin is really powerful. It has very high level of nephrotoxicity. In the long run, it will lead to the kidney failure. And it was, for a long time, a last resort in terms of the treatment. Really lost hope. Well, guess what? There's a plasmid that confers colistin resistance, and it now starts to spread among all the bacteria in the world. It was recently found in the bacteria in the United States. So it's here. It's going to happen. Okay? Daptomycin is a very interesting antimicrobial. I'm not going to ask you about the little intricate details, but what it does is absolutely amazing. So it's a lipopeptide. So it's a part lipid, part peptide. And its lipid part incorporates itself into the membrane. And it's not one molecule, it's many daptomycin molecules. Peptide parts organize in the pore, the channel. And that channel allows the flux of ions across the membrane, bless you. So what happens, cell becomes depolarized, hyperpolarized, whatever. Its membrane potential changes and it screws up cell big time. So it's pretty amazing mechanism, although it works not only on bacteria but on humans as well. So that's why, you know, bacteremia caused by MRSA sounds almost like a death penalty. Okay. So you have to think about what it targets. If it targets membrane, then it's going to be pretty toxic because membranes have a common structure. Does that make sense? Good? Okay.